Hello, my name is Sanin Haveric. The title of this presentation is Toxicity of Depleted Uranium. Naturally occurring uranium isotopes emit alpha particles, positively charged ions consisting of 2 protons and 2 neutrons. Due to their relatively large size and charge, our alpha particles lose kinetic energy quickly and have limited penetrating ability, making them unable to pass through even the outer keratin layer of human skin. As uranium isotopes decay, they transform into other radioactive elements, ultimately leading to stable lead isotopes. This decay process emits beta and gamma radiation. Beta particles can penetrate the skin more effectively than alpha particles, while gamma rays are highly penetrating and pose both internal and external radiation risks. Radon, a radioactive noble gas in the uranium-238 decay chain, readily escapes from soil or rock, significantly contributing to radiation exposure for the global population. Depleted uranium is approximately 3 million times less radioactive than radon and 10 million times less radioactive than americium, which is commonly used in commercial smoke detectors. Depleted uranium has a very long half-life, several hundred million years to several billion years. Most depleted uranium dust particles are reported to be smaller than 5 micrometers, allowing them to stay airborne for extended periods and disperse with wind and water. This dust is typically black, and targets struck by depleted uranium ammunition are often identifiable by a black dust layer on and around the impact area. Studies at United States test sites indicate that most depleted uranium dust settles within 100 meters of the impact site, though some reports suggest that depleted uranium dust can travel up to 40 kilometers and remain airborne for significant periods. When depleted uranium penetrators strike soft targets, such as sand or clay, they are expected to penetrate the ground to depths exceeding 50 centimeters, where they can remain intact for prolonged durations. Impacts on soft targets, such as unarmored vehicles or soil, typically produce minimal dust contamination. This slide shows comparison between natural and depleted uranium radioactivity. Exposure to uranium or depleted uranium is associated with a wide range of adverse health effects, including renal, respiratory, and cardiovascular, including increased risk of kidney disease, diabetes, and hypertension. Health effects from external exposure are limited to skin contact. The dose rate to the skin in the direct contact with a piece of pure depleted uranium is caused by beta particles and is about 2 millisieverts per hour. To exceed the current occupational dose limit for skin of 500 millisieverts, a depleted uranium object would have to stay in direct skin contact for more than 250 hours. Research findings support the conclusion that depleted uranium exposure affects immune system, causing immune dysregulation. However, no single model is sufficient to address the complexity of uranium immunotoxicity. Depleted uranium is involved in inflammatory processes with macrophages as the main target cells. In vivo and in vitro studies have demonstrated that reproductive organs and germ cells were more sensitive to uranium. After exposure, uranium has been detected in the semen and umbilical cord of humans. Evidence for DNA damage, cytogenetic aberrations, mutations, and defects in DNA repair has been reported in miners and residents living in proximity to uranium mine waste sites. Uranium is a bone seeker and readily replaces calcium ions on bone surface and deposits in the skeleton, which occurs in a dose and time-dependent manner. Claims had been made that depleted uranium is likely to cause cancer, leukemia, and other health effects among troops, local populations, and other personnel of humanitarian organizations who were present at locations where depleted uranium ammunition had been used during conflicts, such as Gulf War syndrome, Balkan syndrome, etc. Excess uranium may trigger diverse health problems and toxic effects via oxidative stress, protein interaction, metabolic disorder, cell death, genetic damage, and inflammation. Nevertheless, the toxicological mechanism of uranium remains unclear. Uranium can directly or indirectly interact with the in vivo antioxidant defense system and result in the imbalance of redox homeostasis, which cause impairments of cellular components. Uranium was reported to exhibit pro-oxidant activity in some model systems. Some researchers have shown that uranium can also trigger oxidative stress by impairing mitochondria. In human serum, more than 20% of uranyl ions have been associated with serum proteins. By affecting iron, carbohydrate, osteoblast cell metabolism, vitamin D, dopamine, and cholesterol metabolism, uranium can be responsible for various metabolic disorders. Cell death may be a result of multiple molecular and cellular damages caused by uranium. Uranium also affects caspase 3 and caspase 8 activity in the apoptotic pathway and induces autophagy in osteoblastic cells in vitro. In vivo and in vitro studies have shown that uranium can lead to genomic instability and genotoxicity by increasing chromosome damage, inducing micronuclei formation, interrupting cell cycle progression, and affecting folliculogenesis of and oocyte maturation. Uranium has also been found to affect epigenetic processes. In regards to inflammation, uranium increases expression of cytokines and inflammatory genes. The human body naturally contains approximately 90 micrograms of uranium, and its intake from about 0.7 to 15.3 micrograms per day. In biological fluids, the bioavailable uranium species can be efficiently complexed with bicarbonate, citrate, and proteins. However, different uranyl complexes vary greatly in bioavailability and cytotoxicity. For instance, in vitro toxicological studies have revealed that uranyl citrate was more toxic than uranyl bicarbonate complexes to kidney cells in vitro. Data shows that 2 to 5% of ingested soluble depleted uranium is absorbed into the bloodstream from the intestines, while the remaining 95 to 98 is rapidly excreted. Only about 0.2% of ingested insoluble depleted uranium enters the bloodstream, the absorbed fraction is quickly cleared, with around 90% excreted in urine within the first week after ingestion. The remaining portion is distributed to various tissues and organs, with approximately 10% accumulating in kidneys, where most is eliminated within a few weeks. About 15% deposits in the bones, where uranium remains much longer, and after 5 years a small percentage is still present, and around 1% remains after 25 years. Depleted uranium is a radioactive element that emits high-energy alpha radiation and can create breaks in DNA molecules and form reactive oxygen species that are highly oxidative and source of genetic instability and oxidative stress for cells. Here is presented toxicological mechanism of uranium activity via oxidative stress and DNA damage. While this slide shows how uranium binds to proteins, activates inflammation, or causes apoptosis or autophagy. Thank you for your attention.